So we're going to talk about termination here, and we're going to look at termination in prokaryotes and eukaryotes in the same video. So this is an overview of the three things we're going to talk about. In prokaryotes, there's two types of termination, row-dependent and row-independent, and in eukaryotes, you have rat1 termination. So first, let's talk about row-dependent termination in prokaryotes. Well, what is row? So we have something called row factor, and that's a protein that has helicase activity. If you remember helicase from DNA replication, that's the thing that unwinds the DNA by breaking hydrogen bonds. So if something has helicase activity, that means it can do what helicase can do. It can unwind things and break hydrogen bonds. So row factor can do that. And this is called row-dependent termination, meaning we need row in order to terminate. So there's going to be something called the row utilization site or the RUT site. And this is a sequence that's upstream or ahead of the termination sequence. So if you have your RNA, there's a sequence just kind of in the middle that's called the RUT site. And if row factor is floating around, it's going to see the RUT site and bind. Once it's bound, it can move up the RNA and start going towards your RNA polymerase. However, if RNA polymerase and row were to both keep moving, row would never be able to catch up. So what happens is that after RNA polymerase transcribes the termination sequence, it's going to stop, allowing row factor that binded way over here to catch up. And once it catches up, it uses its helicase activity to unwind the DNA and RNA by breaking those hydrogen bonds, and then your mRNA is released to go do translation. So let's draw what this would look like. So if I have my DNA right here, and I'm only going to draw a single strand for these because technically DNA is double-stranded, but whenever we're doing transcription, we're only worrying about one strand. So I'm just going to draw one strand. So there's going to be a site on this DNA that is going to be the RUT site or the row utilization site. And we also have our termination sequence over here. So this is RUT, and this would be the termination sequence in green. So our RNA polymerase would be moving along. It did RUT, and now it's nearing the termination sequence. So I'm going to draw it all the way over here on our termination sequence with a string of mRNA coming out of it. And on that mRNA, you're going to have that RUT sequence. So our row protein is going to be floating around. It's going to see that RUT sequence, and it's going to bind. And then it's going to travel up your mRNA all the way up to our RNA polymerase, which after it transcribes that termination sequence, it'll pause. And then row will get there, unwind everything, leaving our free mRNA that is happy and ready to go on to translation. But what if we don't have row? In prokaryotes, you also have row-independent termination. And this is also called intrinsic termination because termination is achieved just due to the properties of the RNA sequence and not any external proteins. In this, RNA's polymerase is going to transcribe inverted sequence, and inverted sequence are going to be complementary to each other. And these inverted sequences are going to form a secondary structure because they're complementary, so they're going to want to bind. That's called a hairpin on the mRNA. After that they've bound, you're going to have RNA polymerase is going to continue on, and it's going to transcribe a string of uracil to adenine. So our adenine's on the DNA, and it's going to transcribe a string of uracil. Uracil and adenine only have 2 hydrogen bonds each, and that's not very stable. So we have this hairpin that's here, and then we have this string that's not very stable. So that hairpin is going to destabilize the DNA, and that string of uracil causes the RNA polymerase to pause, and it's not very stable to begin with. It's kind of weak, and then our hairpin is going to destabilize it a bunch, and they're just going to separate. So let's draw what these inverted sequences look like so that makes a little bit more sense. So if I have my DNA right here in blue, and I have sequences on there that are C, 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 G, 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 and then I have a bunch of A's over here. I have my RNA polymerase, and my RNA polymerase is traveling down. So let's say now it is at that point, which means that it's transcribed the stuff before it. So we have that mRNA strand that, if still linear, would look like this: G, 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 because it's complementary, C, 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 C. But G and C really, really, really want to be next to each other. So what's going to happen is this hairpin structure is going to form, where you have G, 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 C, 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 C. So they're complementary, they stick together. And that's unstable for our RNA DNA string together. In addition to that, we have a string of uracil that causes our RNA polymerase to pause. Our adenine and uracil, we just have a bunch of weaker bonds. We have that hairpin forming. Things are not going well for our RNA and DNA. And it's going to form everything, cause everything to detach, leaving that linear mRNA to go on to translation. All right, so that's our two ways that prokaryotes can go through termination: row dependent, where we use row factor that binds to rut site, moves up, uses helicase activity to unwind, or row independent, where it's just due to the sequences, and we'll have a sequence that forms a hairpin, and then that string of uracil to adenine, wherever this destabilizes everything because of the hairpin. We don't like that. And then we have weaker bonds, and everything just attaches. Now let's talk about eukaryotes. And eukaryotes has Rat1 termination. So in eukaryotes, RNA polymerase 2 is a little bit special, and it does not care about termination sequences at all. It'll go up to it, it'll transcribe everything before, it'll transcribe the termination sequence, it'll transcribe afterwards. It doesn't care, it doesn't notice. It'll just transcribe and transcribe and transcribe. So this means if our termination sequence right here, here's our RNA polymerase. It just keeps going. It does not care. And that means if it just keeps going, it's still making a lot of mRNA that has a bunch of unnecessary information that we don't care about. We don't want the rest of this mRNA that's after the termination sequence. RNA polymerase doesn't care about that. So we need some other things that are going to help us out. So if we've transcribed that termination sequence, that means we have a really long string of mRNA. There's going to be some elements that are going to come and look at the mRNA and say, okay, where was it supposed to stop? Where was RNA polymerase 2 supposed to stop? Where's that termination sequence? It's going to find it, cut it, and release that mRNA right here to go do translation. However, RNA polymerase 2 doesn't even notice that the string got cut. It's still going. So we just have a bunch of mRNA that's useless, and RNA polymerase 2 is transcribing even more useless mRNA, which is such a waste of energy. So we have our friend, Rat1, that's going to go attach this extra mRNA and eat it all up like a rat. So it's going to do something called 5' to 3' exonuclease activity. If you remember this from our DNA polymerase 1 in bacteria, 5' to 3' exonuclease means you're removing nucleotides in the 5' to 3' direction. And once Rat1 reaches RNA polymerase 2, it'll make the polymerase detach from the DNA and finally stop transcribing. So let's draw what this looks like. We have our DNA. Here is my termination sequence. And here is my RNA polymerase. He's all the way over here now. He's way past that termination sequence. So if we were to look at the mRNA that he's produced, this is what would match up to be the termination sequence. We only want this little bit right here. That's all we asked RNA polymerase for. But it just went above and beyond, gave us way more than what we asked for. And all of this is just extra, and we really, really don't need it. And we only want this portion right here. So we need a way to get it. So we're going to have some little enzymes come in, and they're going to snip it right here. And they're going to release that piece. So I'm going to draw that. So we have this little piece now that was released, and this is happy and can go on to do translation. But we still have all our extra information that's still hanging out of RNA polymerase 2. And RNA polymerase 2 is still moving. It's making that longer. So we have our lovely little friend, rat1. So rat, those are teeth. And should make the teeth really face that, I guess. Okay, so there's our rat, and it's going to start chopping up all of that extra mRNA. And it's going to move up until it gets to that RNA polymerase. So all that's left behind it are just a bunch of flying chunks of RNA as it's moving up, because it's going chop, 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 chop. It gets to that RNA polymerase, and then it's going to be like, you need to stop, get off the DNA, and then we'll be done. So yeah, that is the last of our termination processes.
I hope you found this video really helpful. The concepts and information presented in these videos will be true no matter what genetics class you are taking. However, the concepts presented in this video are referencing material currently covered in Baylor University's coursework. Remember, if you are currently enrolled as a Baylor student, we offer free tutoring services. Our tutoring center is located on the first floor of the Sid Richardson building. You will find all the details you need to know about these services on our website, www.baylor.edu/tutoring. You may schedule a free 30-minute one-on-one tutoring session online through Navigate, or just drop in during our open business hours. For more information about our current services, please visit our website. Thank you.